Okay, everybody, we're picking back up from where we left off in the last lecture. And just in case you are watching these out of order, the first chapter 3 lecture was all about carbon and how it's like the central atom in a lot of the molecules that we will be looking at and discussing over the rest of the course. So now we're going to move away from carbon by itself and start to focus on something called functional groups. Okay, so this is what a functional group is. It is a specific combination of bonded atoms that always have the same chemical properties, and because they have the same chemical properties, they always react the same way. And it doesn't matter what the carbon skeleton that it's attached to looks like. It's just that these particular molecules, we can guess what they're going to do before they even do it. So that is what a functional group is. There are a total of 1, 2, 3, 4, 5, 6, 7 that you need to be familiar with for this course. So we're going to go through them. We're going to look at them. You should make flashcards. That's the best way, I think, personally, to memorize these. And there's no way around it. You have to memorize these. Okay, so this slide is there because it'll help you recognize how important functional groups are. It can mean the difference between estrogen, which is estradiol, and testosterone. And so for those of you who maybe are like, oh, I've heard of those words, but I don't really know what they are. They're hormones that essentially help a man to become a man and remain a man, and then a woman to do the womanly things that her body also has to do. So without estrogen, puberty, making eggs, having a menstrual cycle, all of those things are out of the window if you don't have estrogen. And then, of course, testosterone is associated with like the facial hair and the taller or bigger body structure for a man. But what is important here is that the rings that are like colored in yellow, those are the same on both molecules. Like if you look at them, they are identical. What is different with these are the functional groups. So it's these like very small highlighted in blue areas on these molecules that make one of them help a woman become a woman and the other help a man to grow into manhood. All right, so here are the 7 functional groups that I was alluding to. So you have hydroxyls, carbonyl, carboxyls, amino, sulfohydryl, phosphate, methyl. And those look like crazy words. They look scary, but they're actually just not. So let's look at each one of them. Okay, this slide I'm not staying on because it is very tiny, but it has all of the chemical groups listed. This is in the e-text. You could definitely Google online functional groups and find something that looks very much like this. But in the next few slides, as the authors always do, they're going to pick apart this graphic and then I'll explain as we move. All right, so they list the first 4 together, and I'm going to skip this one as well so that I can go to the next one, and we can just focus on like the hydroxyl group, then the carbonyl, then the carboxyl, and then the amino group. Okay, so we're at our first functional group. You pronounce this hydroxyl group, and all it is an oxygen atom. Hold on. Okay, an oxygen atom attached to a hydrogen atom. And we already know that like at the other end of this, so like where the oxygen is attached here, you're going to find a carbon. We call these with the OH functional group on them, we call these class of molecules alcohols. They end in OL. So that's a giveaway if you were like reading the name of a molecule and it ended in ol or OL. It's an alcohol, and you know that it'll have the functional group OH attached. the example that they give you is ethanol, and that's one that we're familiar with because humans consume this, and it is in beer, it's in liquor, it's in wine. So this is ethanol. One thing to notice is that the back side of this molecule, so this part here, that is not the functional group, that's actually non-polar. So it doesn't have a charge one way or the other, but when you attach this OH or alcohol group, functional group, to the molecule, it becomes polar. So every time you see an OH attached to a carbon in any of the molecules that we talk about, you know that that particular functional group being present makes that region of the molecule polar. And again, it's polar because just like with water, they have the oxygen that is more electronegative, and then the hydrogen is going to be the partially positive charge. And because it's polar, if you were to put this in an aqueous solution, it is going to, it can dissociate. So extending on this idea, the longer this hydrocarbon chain is, the less soluble the alcohol will become in water. So when it's short like this, like ethanol, it's relatively easy for it to dissociate. The only other thing that you need to remember about this is that it can help with the formation of hydrogen bonding, and it's going to be present in sugars and in some amino acids. So present in carbs and then present in amino acids. The next group is the carbonyl group. So in this particular functional group, you see that there is an oxygen atom and it's double bonded to a carbon atom, and the two areas here could be another carbon. So we have two classes of carbonyl functional groups. You can have ketones and you can have aldehydes. So what is the difference? The ketones, they usually are found within the carbon skeleton. So they're like in the actual molecule. They're not on the outside part. The aldehyde, though, would be on the outside part. But that's the only distinction between these two. So what do you need to know about them? They're also polar. Again, this electronegative oxygen gives it this partially negative charge in that region. Both of these, the ketones and the aldehydes, so carbonyl groups in general, they are present in carbohydrates as well, so present in sugar. And then the only other thing is, just like with the hydroxyl group, the back half of this can be as long as you want to make it. It's just going to depend on whatever the molecule is that you're looking at. And then the same thing goes for these two branches on the sides there and there for the ketone example. And I guess like one way that I kind of remember what a ketone is, I think about a key, it has to fit into a lock, and you know that there is hardware around the key once you put it into the lock. So if you think about that ketone, so being in the dead center, it's kind of like where the key would go in a lock. It fits within the molecule. In this case, it would be where the keyhole would be. All right, so we're moving on to the carboxyl group. What you need to know about carboxyl groups is that they're highly polar, and in an aqueous solution they can ionize, and so what happens is this hydrogen here can pop off, and what you know is that the release of hydrogen ions in a solution can make something become acidic. So carboxylic acid is one of the things that we get when we dissociate the carboxyl group being present in whatever atom you're talking about, not atom, excuse me, whatever molecule you're talking about. So let's highlight the parts like we've done on the others. So you notice that there are 2 oxygen atoms, one here and one here. They both are bound to the carbon. One of them is double bonded, which kind of gives you the whole carbonyl vibe. But the difference is that you have this OH group attached to the carbon. So I already did the oxygen, and there is the hydrogen. And of course, this side, whatever is attached to this particular carbon, it will make a different molecule. So the one example that they give you is acetic acid, which is vinegar. So this is the molecular structure of vinegar, and it has a sour taste. And that's really it. Again, the stuff back here is non-polar. This, however, is highly polar. Highly polar. All right, and then here's a picture of the dissociation or the ionization that has happened on this side of the reaction. So yeah, that's it. All right, amino groups. These are easy to remember. They're the ones with the nitrogen in them. So you have a nitrogen and it's attached to 2 hydrogens. So the blue molecule here, there's your nitrogen, here and here, those are your oxygens. And then again, on the back side of the molecule, we can put any number of different things. So what's interesting here is that there is a carboxyl group present. And I hope that everybody can see it really easily. So here's the oxygen double bound to the carbon, and then that carbon is bonded to an oxygen and a hydrogen. And this particular example atom that we're looking at is called glycine. It is one of the 20 amino acids that we will come across. Let me see. Oh, so they're showing you how it can ionize in an aqueous solution. So you'll notice that you have acid in solution. So this is an acidic environment, but it can be attached to the nitrogen. And that's simply because if you look at the valence of nitrogen, that bond makes sense. Do I want to say anything of note about this one? Let me see. I can tell you that it's polar. Oh, I was writing those in black. Okay. I can tell you that it's polar. I can tell you also that the amine groups, they form hydrogen bonds, and they can be acidic, and they can also be basic. They could be either or. It's really just going to depend on this whole ionization part. All right, so those were the first 4. These are the last 3. So you have the sulfohydryl group, the phosphate group, and then the methyl group. So sulfohydryl group, again, another easy one to remember. It's the only one with sulfur present. So here's your sulfur atom, and here's your hydrogen atom, and on the back side, you can have whatever chain you want. They're referred to as thiols, and here in this molecule, we look at the back half. There should be a number of functional groups that you see here present. You have a carboxyl group here. You've got an amine group here. And then here's your sulfohydryl group there. So what you should see in this particular slide is that there is not necessarily a limit to the number of functional groups that you could spot on a particular molecule. And each of them still carry over their behaviors that we talked about in the previous slides. So whether or not it will dissociate, whether or not it will be acidic or basic, how polar the molecule is, we have to consider all functional groups when we look at that. The main thing that you guys need to know about the sulfohydryl groups is that they're present in some of the amino acids. This one example that they're giving, cysteine, it's a sulfur-containing amino acid. So there you go with that. And then also, they're able to form something called disulfide bonds. And when we get a little bit further along in this chapter, those bonds come up. Keep going. This is a phosphate group. So this large molecule in the center is phosphorus. It's surrounded by 4 oxygen atoms. Notice that the oxygens are carrying a negative charge. This has to deal with the distribution of the electrons around this atom. Let me see. Oh, so they're saying they're called organic phosphate, which is not a huge thing in and of itself. And the particular one that they're showing you here, glycerol phosphate, it's important for a number of different chemical reactions. Besides that, all you need to know is that the phosphate group itself is polar. It's polar because of those oxygen atoms again. It's acidic. So how is it acidic? Because when it loses hydrogens in solution, and I'm not talking about like these ones down here, these are pretty stable. But when you look at some of these OH groups here, and then also the fact that this has a negative charge, it would be willing to take some, not take some, but it would be willing to pop off some hydrogens if there were some present into solution. What else do I want to say about this? That's really it. This particular group is really important in ATP, which is what you need for energy to get chemical reactions going in the body. So ATP. All right, the last group is the methyl group. And methyl groups are just a carbon bound to 3 hydrogens. And so we see here the carbon atom, here are the 3 hydrogen atoms. They call these methylated compounds when you stick a methyl group onto a molecule. In this case, we're looking at something called 5-methylcytosine. This is a part of DNA. It's been changed slightly because of the addition of this methyl group, so it's been methylated. But let's see if you guys can guess why this is called 5-methylcytosine. You should pause me for a second and then attempt to count the carbons to see if you can come up with where we start counting and how we end up at 5. Okay, so the counting on this is not as intuitive as it may seem, but this is what I'd like to point out to you all. So if you start by just looking at the fact that this is a methylated compound, meaning there has been a methyl group or a methyl functional group added to the cytosine structure, what you'll find is that each of the carbons around the ring already have something attached to them. The exception would be like this methyl group right here. So like this by itself, like now that molecule that you're looking at, that is cytosine. But 5-methylcytosine lets me know that whatever carbon is open is the one that's going to be the methylated carbon. In this case, if we do the count, what you'll see is that you get a numbering like this. So this one here, the first carbon is already taken up, so that's carbon 1. It's being paired, double bonded with the carbon, it's bonded to a nitrogen, it's bonded to a hydrogen. The second carbon position has it's double bonded to an O, which is a carbonyl. So there's a carbonyl group for the second one. The next one, the next carbon is carbon number 3. That one has an amine group. Hold on. I don't know why the 3 didn't draw there. Yeah, so that has the NH2, so it has amino group on it. And then you get to the fourth one here, this fourth carbon. And there's nothing attached to it normally. So once you find that one, the methyl group that you add on to that carbon would be counted as carbon number 5. The nomenclature, which means like naming rules for organic molecules, can get like complicated very quickly. So you're not really expected to memorize, you know, like 5-methylcytosine. But what you should know is that if a regular cytosine ring it's not methylated, and then I'm telling you that it is methylated, you do have to be able to find the spot that is open that can receive the methyl group. So, I mean, like, that's it. Those are all of the functional groups. And the last slide for this part of it is dedicated to talking about adenosine triphosphate, or ATP. And the only thing that you really need to know is that you have an adenine group, which is going to be part of one of the amino acids that we see in the protein part of this chapter. The tri means 3, so TRI is 3, and the P being a phosphate, you know what a phosphate group is. It's the phosphate surrounded by the 3 oxygens. That is what ATP is. So you'll notice that the names give a general description of what the molecule will be composed of. The way that ATP's energy is harnessed is that the bonds that keep one phosphate group linked to another, when they are broken, you get a release of energy. And when we go into metabolism, photosynthesis, and cellular respiration, the chemical reaction that this particular atom would take part in, you will see, and you'll understand why I call it like the energy coin or the energy currency. It's like the money that keeps the energy going. Okay, so that's it for functional groups. Again, make flashcards, write down what's polar, draw the structures. I encourage you also to maybe try to find different functional groups on your own, like for practice. I do have a worksheet that goes with this. I know even day classes already have it. If you're an odd day class, you were not given the sheet as of yet, but it's coming. So that's it, guys. See you on the next one.